I have selected some questions that we frequently get and will present today, but as Gunnar said, it's also possible to ask questions related to EUCAST AST methods, even if it's not related to the subjects that I am speaking about today. There is a specific heading for frequently asked questions on the website in the left-hand margin of the start page. So if you go there, you will come into a document where we have collected a lot of questions that we have gotten through the years. The questions are divided into different subheadings to easily navigate, and all questions are also linked to the answer. So you can click on the link here to get to the answer. And it might look like this, for example. And the document is updated usually once a year. And we add more questions and make sure that the answers are still accurate, depending on breakpoint changes, etc. There are breakpoints related.
And particularly this disease program has been doing with providing a
Gunnar, do you have your microphone on?
in the EUEA.
Sorry?
And then we'll move on to your collaboration with WHO.
Sorry, Erika, what was that?
No, it was just someone is having the microphone on with some background.
I'll mute again.
Thank you. So there are questions on AST methods and also quite a lot of questions on breakpoints in this document. But today I will only focus on AST method related questions. And the first one is a quite specific question, and it's about benzyl penicillin MIC testing in Streptococcus pneumonia. And the question is that for isolates that are screen positive or resistant with the oxacillin 1 microgram discs diffusion test, if we do a benzyl penicillin MIC that is susceptible according to the breakpoint, which result should we trust? The oxacillin 1 microgram screening test is a very sensitive test to detect beta-lactam resistance mechanisms in Streptococcus pneumoniae. The screening breakpoint is the same as the ECOFF. It's less than 20 millimeters, and it means that isolates that have black bars here and belong to the wild type, they do not have beta-lactam resistance mechanisms, whereas isolates with zone limiters less than 20 millimeters have beta-lactam resistance mechanisms, and they are resistant to one or several beta-lactam agents. If we look at benzyl penicillin specifically, this is how oxacillin 1 microgram zone diameter sizes correlates to benzyl penicillin MICs. These MICs are all with broth microdilution. The colors of the bars correspond to different MIC values, and they correspond to the clinical breakpoints. And here I have used the breakpoints for meningitis, which is S less than or equal to 0.06, resistant more than 0.06 milligrams per liter. So the green shades here belong to susceptible results, and orange and red to resistant results. You can see also here that we have tested many isolates with beta-lactam resistance mechanisms, of which some have very high benzyl penicillin MICs and less wild-type isolates. So this is a collection where we have provoked the system with many different resistance levels for beta-lactam resistance. And you can see also that below the screening breakpoint of 20 millimeters, there are a few isolates that actually test susceptible to benzyl penicillin with broth microdilution. And as long as you perform broth microdilution, you can trust these results. And when your quality control for benzyl penicillin is fine, these isolates that are below 20 millimeters for oxacillin and test susceptible for benzyl penicillin, they have beta-lactam resistance mechanisms, but it doesn't affect benzyl penicillin in this case. We know that these isolates are resistant to phenoxymethylpenicillin. But we also know that many labs don't perform broth microdilution for Streptococcus pneumoniae or other organisms, and the most commonly used method for this is gradient tests. And we therefore decided to look into specifically gradient tests for benzyl penicillin MIC testing in Streptococcus pneumoniae. And we have selected a collection of 57 isolates, so not so many, but many of them with beta-lactam resistance mechanisms. And 45 of these had benzyl penicillin MICs above 0.06 milligrams per liter. And you can see in the graph there how they distribute, and also the different breakpoints that we have. The meningitis breakpoint, which is less than or equal to 0.06, more than 0.06, and then the breakpoints for other infections than meningitis, where you have a resistant breakpoint of more than 2 milligrams per liter. We looked into 2 brands of gradient tests, E-test from bioMérieux and MTS from Leeuwenhoek. We used MHF plates that had been prepared from BBL and oxoid Miller-Hinton agar to see if we also had some specific problems related to the Miller-Hinton agar used. This is what it looks like when we correlate the MICs for gradient tests to broth microdilution. You have E-test in the upper part here and MTS in the lower part. The reference broth micro MICs are in columns and the MICs for the gradient tests in rows. The different breakpoints are shown as lines. And if it's a green box here, it means that the broth microdilution MIC and the gradient test MIC are identical. And the yellow boxes show MICs that are 1 dilution higher or lower with the gradient test compared to broth microdilution MIC. So actually most of the, quite a large proportion of the MICs are within ±1 dilution of the broth micro MIC. But we can also see that there is a clear tendency that MICs are lower with gradient tests. And if we look at the proportions, you can see that the number of results below the broth micro MICs are much higher than the ones above the broth micro MIC. And when we come close to the breakpoints, for example, MICs as 0.1 to 5 and MICs at 4, we also see that we get categorical errors when we test with gradient tests. So both E-test and MTS had systematically lower benzyl penicillin MICs for Streptococcus pneumoniae compared to broth microdilution. And if you put all the data together, both brands of gradient tests, both media used, as much as 60.5% of the MICs were below the broth micro MICs, usually 1 to 3 dilutions, and only 3.5% above the MICs. There were some differences related to brands of gradient tests and agar, but categorical errors were observed for all this combination. And the major correlation that we saw here was that the gradient tests systematically underestimated MICs. Based on this, we have published a warning on the EUCAST website. If you go to AST of bacteria and warnings, you will find the current warnings that we have. To warn against that E-test and MTS systematically underestimate benzyl penicillin MICs in Streptococcus pneumoniae. Our recommendation is to first trust the oxacillin 1 microgram screening test. If it is screen positive, this isolate have beta-lactam resistance mechanisms, and you should interpret gradient test MICs with care. Also, for isolates that test at 0.5 to 2 milligrams per liter with gradient tests, we recommend that those should be checked with broth microdilution to get a reliable MIC. To help laboratories to validate or evaluate MIC methods for Streptococcus pneumoniae, we have put together a collection of 10 isolates with varying beta-lactam susceptibility, together with the Swedish culture collection CCUG. And this strain collection can be ordered from CCUG as a package of 10 isolates. And it's focused on MIC determination of benzyl penicillin, but reference values are also available for several other agents and also agents from other classes. Reference MICs are available on the EUCAST website, and there you can also find information on how to order this strain collection. The next question relates to organisms which are yet not covered by the current EUCAST recommendations. We are constantly working with adding criteria for more species, more organisms to the EUCAST system. And this is what we have added since 2012. And the newest additions for this year are Achromobacter and Bacillus species. What we are currently working with are Nocardia species. For Nocardia, we will recommend disc diffusion on the Millihinton fastidius MHF agar and also agar dilution on the same media as the reference method. Nocardia grow in broth, but they grow much poorer and it doesn't really add much to add the lysed horse blood to the broth. You get a much more reliable result if you use agar dilution. For Vibrio, we have also started to test it. That will be just according to standard broth microdilution, unsupplemented broth, and standard disc diffusion on unsupplemented Millihinton agar. We do have methods and breakpoints for Corynebacterium species, but not for Corynebacterium diphtheria. So we are also coordinating a project where 2 external laboratories are testing Corynebacteria diphtheria according to the EUCAST method, which is Millihinton F, Millihinton fastidius broth, and MHF agar plates. And finally, we are also developing a disc diffusion method of rapidly growing anaerobes, and for that we will recommend fastidius anaerobe agar FAA. The anaerobes that the method have been developed for are Bacteroides, Prevotella, Fusobacterium necrophorum, Clostridium perfringens, and Cutibacterium acnes. So it cannot be used, so far it cannot be used for any other species. So we have looked at these 5 species and also at antimicrobial agents that are most important. The media will be fastidius anaerobe agar with 5% defibrinated horse blood, an inoculum McFarland 1. Incubation in an anaerobic environment for 16 to 20 hours. And the species that we have tested, they grow well within 16 to 20 hours on this media, and prolonged incubation is not necessary, and it's also not permitted since it will result in different zone sizes. Also for anaerobes, we use agar dilution as the reference method, and we have just published our method development for this method in CMI, and this paper is available online on the CMI website. We are currently performing a multi-laboratory trial with 16 laboratories, where these 16 labs in different European countries will test a collection of 38 isolates belonging to these 5 species that we have put together, and then we will look at reproducibility and robustness of the method, and then finally be able to publish the method and also preliminary zone diameter breakpoints. Here's an example for the anaerobes and meropenem for different organisms and species here. The first one is for Bacteroides, where we do have non-wild type isolates and isolates that have MICs that are either in the I category or resistant. And you can see here that these are nicely separated from the wild type distribution, which is in the green bars here from 28 millimeters and above. For the other species, all isolates are susceptible to meropenem, but if you look at in the lower right-hand part of the slide, you can see that, for example, for Clostridium perfringens, isolates with MICs of 0.1 to 5, they are in the lower part of the wild type and just below wild type distribution. So we still see that we have a very good correlation between inhibition zones and MICs. As part of this multi-laboratory trial, the labs will also then test local clinical isolates, and we hope to be able to collect a few more resistant isolates to provoke our system a little bit more when we set the breakpoints. A question regarding the MHF plates, Millihinton fastidius media. We know that some laboratories have problems with too excess humidity in MHF plates. The main problem with this is that it might cause fuzzy zone edges or haze within zones that makes it more difficult to read the zones, and the zones can also be smaller than they should be due to this haze and fuzziness. First of all, it's important that before you inoculate your plates, and this is the same for all disc diffusion testing, that your agar plates are at room temperature and have a dry surface. There shouldn't be any drops of water on the surface or inside the lid. If you have problems with condensation like this, or if you have problems with fuzzy zone edges or haze within zone, we recommend that you dry the plates prior to inoculation, and you can also look into how you handle and store your plate to reduce this problem. If you prepare plates in-house, we recommend that when they are newly produced the same day of production, that they are left in room temperature overnight to remove excess humidity before you put them in the fridge. If you use plates that are stored in plastic bags or sealed containers, you can unpack the plates that you expect to use within a week or so and store them unpacked in the fridge. And then we recommend that you also dry the plates in room temperature or in an incubator before inoculation. And this can actually have a quite large effect on the zone edges and make it easier to read the zone the next day. So when you dry plates, you can either do that at room temperature, 20 to 25 degrees overnight, and then you just have the plates in stacks, or you put them in the incubator at 35 degrees Celsius with the lid removed for approximately 15 minutes. We also get a lot of questions on quality control problems. How should we troubleshoot? How should we perform quality control? What does the target values mean, etc. So I will also go a little bit into this. This is a part of the EUCAST QC table. And as you can see, both for MICs and inhibition zone diameter, we have both ranges and target values. The ranges is the acceptable limit, the lowest and highest acceptable value that you should have for this specific strain agent combination. And ranges are always set to allow for random variation. The target values, that is the middle of the range, and that is what we are aiming for. When you have repeated measurements, your target for disc diffusion or your mean value should optimally be on target plus minus 1 millimeter and your mode MIC on the target value. We know that might not be possible for every strain agent combination that you test, but we also know that it's possible to end up close to the target value for most of the strain agent combinations that you test. And it's also a very good way to troubleshoot if you have problems with a specific agent, or if you, for example, have many results where you are above the target value, then it might be related to the agar, to the reading, etc. So it helps troubleshoot when and understand what affects the zone sizes. This is an example also what shows why we have the meaning of the range. This is E. coli Atcc 25922, piperacillin tazobactam, and in the graph to the left, we have taken 20 discs from the same cartridge. We put it on Miller-Hinton agar plate inoculated with the same inoculum. They were of course put into the incubator at the same time, and one reader read all the zones. And then you can see that almost all results were at 23 millimeters, a few at 24 millimeters. So these discs had a very good reproducibility and were reliable within one disc cartridge. And they also have a content where you get a zone in the middle of the range and close to the target value. If we then take discs from the same manufacturer, but different disc lots and put it into our routine testing, we have different lots on Miller-Hinton agar, different lots of discs, 200 test occasions and 10 to 15 readers. Then you see that we still have most of our results at 23, 24 and 25 millimeters. So we are close to the target value, and then we have a fewer and fewer isolates close to the limits. So routine QC data must allow for some variation, but it's important that the disc potency is correct from the beginning so that you get into the middle of the range and not close to the lower or upper limit. So the aim here is to have mean values close to the target value, and we know that we have to allow for some variation, day-to-day variation due to small differences in inoculum preparation, plate inoculation, incubation, time and temperature, and reading of results. We also recommend that you look at your QC data over time. And this is an example where zones have been measured once a day for 40 days. So each dot here is an individual zone diameter. You see the upper and lower limits and also the target value in the middle. Sometimes you will get a single result out on range. And our first recommendation is then to just exclude any obvious reason for that. If you get a zone that is too large and your inoculum for the QC strain is too low, that is the most probable explanation. Then you can just repeat the test and look at the results the next day. If you get a zone that is too small, you can again look at the inoculum. Is this too heavy? Then it might be the inoculum again. It might also be that the antibiotic discs have lost potency, and you can just start over with a new cartridge. As soon as we open a cartridge of disc, the discs are sensitive to degradation due to the humidity in the air, and discs don't really like to be moved between fridge and room temperature, so they should always be at room temperature when you open a cartridge or a dispenser. Another thing you can detect if you analyze your data like this is what happens at day 30. Then your zones are starting to get smaller. This might be due to degradation of the antibiotic agent. And in the end here, you have 2 consecutive results that are out of range on the same side of the target. If you're observant here, you can detect this problem already before you have results out of range because you see that your zones are getting smaller. And again, the first thing you can try is just to open a new disc cartridge and of course check if your discs have passed expiry date and if you need to start using another batch of discs. Here's an example where I have just kept the readings at day 7, 14, 21, 28, and 35. So once a week QC. And then you can see that everything looks pretty nice, but we know that we had results out of range. And also when we test only once a week, it's quite difficult to pick up this trend when we are getting decreasing zone sizes. So this is the reason why EUCAST recommends daily QC or at least 4 times a week. It's because that's the way you can detect problems before and avoid reporting erroneous results for clinical isolates. Is it then possible to have results like this? Yes, it is, I would say. This is again an example from our routine lab. 2.5 years of QC testing for Staph aureus Atcc 29213 with cefoxitin 30 microgram disc. And you can see that we have a few readings out of range, but most of them are within the range. They are sometimes closer to the upper, sometimes closer to the lower limit. But if we look at the mean value here, it's close to the target value. Another way to quality control your data is to compare distributions, your own local distributions with distributions in the EUCAST reference database. This is a very good way to detect systematic deviations, and it's actually sometimes more sensitive than testing with quality control strains. Also, when laboratories contact us and say that they have problem with the specific strain agent combination, we always ask if they have data for routine isolates, zone diameters, or MICs that we can compare with the reference distributions. This is where you will find the database on the EUCAST website. And you can do it both for MIC and for distribution. So first 2 examples with MICs. What you do when you compare with the reference distributions is that you look only at the wild type distribution. And this means that it has to be a test that you perform consecutively or primary testing. It cannot be a follow-up test that you have for gram multi-resistant bacteria, for example, because then it will be totally biased towards resistance. But as long as you have a sufficient number of wild type or just normal isolates here, you can perform this comparison. The first example here is for Staph aureus with gentamicin. This is the reference distribution. The median is probably in between 0.25 and 0.5, but with the highest bar at 0.5. If we look at our routine testing for this combination, it's quite similar, and we have our highest bar and our median at 0.5 milligrams per liter. Second example is for Streptococcus pneumonia with levofloxacin, a very clear median of 1 milligrams per liter, and also in our testing. So here we have two combinations of organisms and antibiotic agents where we have a very good correlation and a well-calibrated test. And of course, you can do the same with disc diffusion. This is an example with E. coli and cefotaxime 5 microgram disc. First, the reference distribution, where we have a median at 28 millimeters. If we look at data from another lab, which we can call lab A, they also have a median at 28 millimeters. Their distribution is a little bit tighter, which is to be expected since this is only one lab, and the reference distribution contains data from many labs and with different manufacturers of agar and discs. Lab B, on the other hand, they have a systematic deviation with their median being 2 millimeters larger. Their median is at 30 millimeter. The red line here is still at 28. And the risk with having a systematic deviation like this is that you will report probably some isolates as false susceptible. And if it looks like this, we recommend that you troubleshoot and try to understand why you have a deviation like this. And when you troubleshoot, there are a number of potential sources of errors, but you can always start with something that is easy to correct and easy to investigate, and then look into more and more detail. So usually we always start with looking at the discs, storage and handling. That should be according to the manufacturer's instructions. The discs are sensitive to degradation, as I said, or the antibiotic in the discs. Check the expiry date. Sometimes it might be the wrong disc potency that has been used, or actually even the wrong agent. So sometimes you can find an easy explanation here. Media could also be a factor, storage and handling. Of course, the Millihinton agar base, from which manufacturer you buy it. Again, expiry date, supplements, are they correct? Are they of good quality? The excess humidity we already talked about, and also there should be a specific agar depth to get correct results. The next thing to look into could be the procedure, how you prepare your inoculum, the measurement of the McFarlane value, how you inoculate plate, incubation, is it the correct time, atmosphere and temperature, and also reading of results. And also finally, sometimes out of range QC results might be due to the control strains. It's also very important to store and handle control strains properly and according to the manufacturer's instructions. There are also some instructions in the EUCAST distribution manual on this. Strains can be too old and they can get mutated, and some resistant strains might lose their resistance if it's on a plasmid, for example. And we are very happy to help, to troubleshoot. And usually I would say that we get the question. Sometimes we probably have a clue or a quite clear answer where the problem is. Sometimes we have to troubleshoot together with the laboratory, and usually we are able to find out the reason for the deviating results. So you are always welcome to contact us with any questions you have relating quality control and the EUCAST AST methods. So I can see the chat function here while I am sharing my screen. So Gunnar, are there any questions in the chat, or does anyone have a question? Okay, I am now looking into the chat.
There's a detailed question from Sebastian.
Yes, that might also be a bit your area, John. But yeah, we could say that the question is, or if a QC range, for example, for an MIC range is usually 3 dilutions. A target value plus minus 1 dilution. But then when we look at the wild type distributions, those are quite often a range over 5 dilution. And why is it like that? Here comes an example. Is it Gunnar? Are you sharing the screen?
Yeah, you go ahead.
Yes, so here you can see in the upper part of the graph, we have Streptococcus pneumoniae tetracycline. The first graph to the left is the QC strain, quite many repeats. And then we have clinical isolates with the same agents. And then both are from our EDL testing and then the reference distribution from the EUCAST website. So quite similar to what we looked at before. And then you can see again that when we test clinical isolates, we have more variation. And we think that one reason for that is that these isolates have different growth characteristics. Some of them grow faster and some of them grow slower, but the QC strain behaves more similar at each test occasion. So that is one of the explanations. And then there are probably a number of factors that affect, or that we could call biological variation. So we add biological variation when we test clinical isolates.
Sorry, may I ask another question? So I totally understand that for a population of bacteria. Now, if we retest the same clinical isolates multiple times, do we still observe this 5 dilution range of variability? And why then? Because
do
it's
you
the same
mean
strain.
if you, I mean, either if you test one QC strain 100 times, and if you test one clinical isolate 100 times, you probably get the variation is probably quite similar if the clinical isolate is stable and it has the same, it's not affected that much in its growth characteristics, because the QC strains are also selected to be stable strains. But if you test different isolates, then you will add that biological variation, that some of them grow faster and some of them grow slower.
Okay, thanks.
I'd have to add, Erika, that if you have to add that unless you do it an extremely large number of times, you will see variation between different strains. Some are only going over 2 dilutions, but even though there's 20 or 30 replicates. So it isn't, it's a noisy issue, it isn't simple. And we can only assume, because nobody's done the experiments, that when you've done it 100 times, they all kind of resemble each other.
And is there anything to do to increase the accuracy when you test one strain or one isolate multiple times?
Nope.
Yeah, unless you can, of course, if you can minimize the factors that might cause variation. If the same person is reading all the tests, you will have probably a little less variation than if different persons read the same test. We try to standardize reading as much as possible, but that is one part of the AST that is most difficult to standardize. We believe that there is a wrong way and a right way to read MICs and zone diameters, but still some zones are fuzzy, some MICs have some trailing endpoints, and it's not always easy to read the same. But of course, if the same person reads all the results, your variation will probably be smaller than if different persons read the MICs.
But I think an important point here is that as soon as you try to reduce the noise, you will also introduce a systematic bias, because the only way to reduce the noise is to decide that I will only use Muir-Hinton from this company and I will only use this particular batch, because otherwise I will have some noise from the fact that I'm use different batches, or I will have to use only discs from this vial and only from one company and only from a batch that was produced in January 2015 or whatever. So whenever you do that, you will introduce a systematic bias into your results, and then you can reduce the noise somewhat. You cannot do away with it, but you can reduce the noise and the random variation somewhat. But from a practical point of view, you're digging yourself a deep hole from which you cannot extract yourself, because then you're sort of doomed to use that company's products for all eternity and in all discussions for the future. So you have to allow for some random variation, and you need to avoid systematic bias, if you like.
And we can also add that we also have to allow for reasonable small variations between materials from different manufacturers, but those should be within reasonable limits. And when we set QC criteria and when we develop breakpoints, we always include these variations in our data sets. So we have media from different manufacturers, we have discs from different manufacturers, we have different readers, etc. So, and tested at different days. So the incubation time will not be exactly 18 hours every time, it will be between 16 and 20 hours. So that variation is already built into the EUCAST system and is included in the QC ranges and the breakpoints that we set.
To return to the question, is there a difference between an Atcc strain and a, let's say, a clinical strain? I think the principal answer is that no, there isn't a difference, but there are some clinical strains from some patients with chronic infections like cystic fibrosis, and I'm sure we can come up with a number of other chronic infections, where the strain has been attenuated by the situation in which it has been living. And that will probably behave somewhat different. But if you take sort of the run-of-the-mill E. coli from urinary tract infections from women, or men for that matter, and you pick a random one and you repeat it 100 times, at the same time as you repeat an Atcc strain 100 times, you are going to find a very similar distribution over the results. It may differ between different agents because different agents have slightly different properties when it comes to how easy they are to read, how they react with the media, etc., etc. But basically all of that will hit both the clinical strain and the Atcc strain. So this is what we have, and this is what we have to live with. And I can see no reason, no way of reducing that random variation unless I'm prepared to introduce a systematic bias in the system.
Thank you.
Can I say that it was a bit unfortunate at the very beginning because some people used the link from this morning and it was a different link, as you discovered eventually, for this afternoon. The reason we have different links for the morning and afternoon, and that was the same during the autumn webinars, 5 of those, is that it affects the recording of the webinar. We want to have individual recordings of each of the webinars, and then this is the way we do it. So I apologize for that. And the only consolation is that there will be an excellent recording of the whole presentation, both Erika's and John's, available on the website from probably tonight at 2400 or something. And for anyone who missed the first part of what Erika was saying, you can go back and have a look at that there. So again, apologies for that. I could have made it more clear in my announcement, and I will do that next time. So with that, and unless someone wants to say something about that, we will go on. No more questions for Eric at this time, sorry. Then I invite John to talk about ECOFFinder and how to use ECOFFinder. And you should be able to share your screen, John.
Yes, thank you. Thank you, everybody. And yeah, I'm sorry, even I got confused because it was the first time I've joined both meetings. So at least I was able to guide the people on the other meeting to where the right place to go was. I'm going to try to show you how to use ECOFFinder which is a, where are you? Somehow I'm struggling to find my link. There we are. Thank you. And I'm going to assume that most of you know what ECOFFinder is. It's freely available on the website. It is for assisting in the definition of ECOFFs. And ECOFFs, as you know, are in a sense, the guide to the end of the wild type distribution, and are used to distinguish phenotypes, in other words, wild type phenotypes, from those with acquired and phenotypically expressed mechanisms. So what is ECOFFinder? It's actually an Excel workbook. It's macro enabled to undertake a few analytical functions, and it can be used to estimate ECOFFs from any MIC distribution. It doesn't care if it's a bacterium or a fungus or a virus or a humans. If you've got a minimum inhibitory concentration or something similar, you could use it. It's freely available on the website. The link is there. But it won't take you much, even a Google search. Just put EUCAST and ECOFFinder, you'll find it. It is not locked down. There are no sheets, there are no cells, there are no pages that are locked down. And so it is easy to modify. It's quite deliberate. People can use the content the way they choose. But because it's not locked down, always keep a separate copy that you're not using, because it's easy to accidentally corrupt, like delete a formula somewhere, which can have effects across the whole of the worksheets. After many years, it finally works both on PCs and Macs, because for a while, the solve function on the analysis worksheet wouldn't work on a Mac. But fortunately, complaint to the supplier to Microsoft finally had an impact, and that's been fixed. I'm just showing you here one of the sheets on ECOFFinder, is just to remind you about who to contact. That's namely me if you have particular questions about it. There's my email addresses, and it's also got the reference to how ECOFFinder came about. So ECOFFinder is there to provide a mathematical analysis of the data that we actually have. Many of you would be aware that ECOFF's been around for a very long time, ever since the MIC distribution website was created. And most of the ECOFFs that you will see and currently see have been determined by the very keen and knowledgeable eye of gynecolometer. So it's an eyeball technique. It works pretty well because it relies on the fact that when we look at MIC distributions and we see wild types, we see something symmetrical, and it works very well. But I decided that we at least needed some kind of mathematical support for that. Came up with this idea of iteratively fitting a cumulative log normal distribution. Why log normal? Well, MICs are distributed on a log2 scale, and you've been looking at log normal distributions on these graphs since forever without actually recognizing it, because the x-axis is logarithmic. What ECOFFinder does is take repeated subsets of the MIC distribution, such as the one distributed here, and analyze them to see if it can fit a cumulative log normal distribution. So it'll start with one beyond the mode in the wild type and analyze that, and then it'll try the next one, 2 above the mode, 3 above the mode, and so on, until it finds what I would call a best fit, where the number in the wild type distribution most closely resembles the number actually seen in that distribution at that MIC. You can read the paper if you want more information. For those of you who have used ECOFFinder, you already know this because you may have made this error. It's not an error, it's just a trick that you need to actually activate an add-in that comes with every version of Excel called Solver. And you won't be able to press the critical button and get sensible results. You'll usually get an error message. So this is the Solver. It comes with every version of Excel, but it does need to be activated. You'll find how to activate it in File, Options, Add-ins, Manage Excel Add-ins, Go and tick and Solver Add-in. I'll show you that graphically in a moment. And as I said, unless Solver is activated, clicking the Solve button on the analysis spreadsheet will result in an error. So this is the current version of Excel that I'm using. It's Microsoft 365, but it has been looking like this for a few years at least, and a few versions. And if you go to Excel Options, you will find a menu on the left. Here are add-ins. The main screen, you will see the add-ins that are currently operating. You can see what's operating on my version of Excel. And the Solver is already added in on mine, of course, but if it isn't on yours, you don't see it on this active list here. You go to Manage Excel Add-ins and select Go, and you will get this menu, and you'll find a blank against Solver add-in, which if you tick, will change to added in, and it will stay added in your version of Excel until you switch it off, whether you're using ECOFFinder or not. It doesn't matter that it's sitting there, it won't cause any harm. Now to ECOFFinder itself, there are 7 worksheets. One just shown before is about ECOFFinder, just information about the version, who to contact, namely me, and also what the source of the analytical technique is, the reference. There's a tab on changes from the previous version. That's fairly obvious for every version change. There's an instructions sheet for those new users who aren't familiar at all with how ECOFFinder might work. Then the next 2 sheets, data entry and analysis, are the main work area. They both have separate functions. Data entry, both where you put the data in, but also where the results are displayed. And the analysis worksheet, where all the number crunching actually happens, where the iterative process happens. There's a sheet with demo data, which we'll use to show you quite soon. If you're a new user, you can get to play with it to make sure it does what you think it should do. And finally, a recent add-in feature of version 2 versus version 1 was a results summary, where if you click a certain button on the data entry sheet, the results that you've generated will be stored for later use if you so wish. So here's just a quick overview of those sheets. This is the information currently sitting on version 2.1 of the current changes from the previous version. As you can see, there were quite a number of changes, even from version 2 to version 2.1. They were small changes, but there were quite a lot of little ones. A detailed view, unreadable to you, I'm sure, of the ECOFFinder instructions. What you will not see on the version you have downloaded from the website is this black box warning here. It was clearly an oversight on my part not to put this here because it's one of the commonest questions I get. I clicked on the Solver button, I got an error message, what did I do wrong? And the answer was, you did nothing wrong, you just didn't know to activate the Solver add-in. So that will be corrected in version 2.2 or 2.3 or higher, whatever number we decide needs to be the next one. This is a view of the data entry sheet. for your visualization, I've added some data from one of the demo data, so Strep pneumonia and ciprofloxacin. I think there are about 60-something distributions when this was data was accessed from an older version of the MIC database. And you can see in this green column where the raw data has been entered, the rest of the data that you can see before the analysis has actually happened are sitting here. There's not much. You can see all these funny Excel squiggles, which means nothing makes sense until the calculations have happened. But most importantly, you'll at least be able to visualize the data you've entered. A quick word of warning, I wish I'd said this morning, was it's very easy to paste data into the wrong spot. So you've got to be really careful that when you do paste the data, you line it up with the appropriate MIC. The other really critically important tab is the analysis tab. And I'll spend a little bit of time here because it looks very complicated. It's not complicated for a user, it's just complicated to look at. A lot of the stuff at the bottom of the screen here is redundant. It's actually just used so that the results can be extracted back to the data entry sheet. So having entered the data, ECOFFinder makes a decision about where the mode is, and usually that's in the middle of the wild type somewhere, and it gives the log2 value of that. The log2 value is chosen because it actually is easier to manage subsequently. It would be a rather complicated Excel formula to try and put it into other values, but just for your purposes, the actual value is displayed underneath. And then there's this, then there's this area called starting values. In order to make the iterative process work and solver to work, you need some starting values. Solver works by entering some starting values and iteratively playing with the numbers until you get a fit. You need to add starting values for the number you think is in the wild type. And usually you can get an idea by that by looking at these numbers here, and this one looks, maybe it's 65,000 or something around there. Then you need to enter a guesstimate for the log2 mean. And you can start with the mode, that seems to be the simplest, and then an estimate for the log2 standard deviation, which is usually somewhere between 0.5 and 1, and if you start with a value 0.7, you will usually not go wrong. And then you need to drag those values all the way through to wherever you see data displayed above, which is where there are actual values in the distribution. And once that's happened, you click solve and some magic happens over a period of some seconds, and the data are restored back to the data entry sheet, the actual answers are available there. Here's the data on the demonstration data sheet. We will use a couple of those just to show you how things work. And I see I've even then haven't corrected my error. The name change to Klebsiella aerogenes has caught me out there. And finally, the results summary sheet, where I actually have run that Streptococcus pneumoniae ciprofloxacin data, and the results are actually stored on this sheet for later use if you so happen to want to do that. There's another way of saving the results, and that's to click this button called Copy Results Image, and that will create this image, which is literally what I did to paste this onto a PowerPoint slide. And so you've got a permanent visual record, which also includes your opportunity to look at the MIC distribution graph for later reference. So just a reminder, ECOFFinder 2.1 is sitting here, and the link is on the EUCAST website. And now what I plan to do is show you a couple of examples using the demonstration data that we had before. And here we go. Here's a blank ECOFFinder version 2.1. And we will start by entering data, putting the name of the organism. You don't have to put this information here, but if you wanted to store the data, these are the labels for the bug and the drug that you actually studied. You may actually know the number of distributions involved. I think for this one was 60 something. I can't remember, it was quite a while ago. And I will then go and get the data, which are sitting on the data thing, copy that information and paste it into here. And here you can see that graph that I showed you before. There are some results sitting here already, but the results that you're really interested in are this ECOFF box right in the middle. And you won't get those results until you go to the analysis sheet. And in order to run the analysis, you need to choose some values. We discussed before, something like, there might be something like 65,000 values there. We thought for a log2 starting mean, we might as well use the mode in this darker blue box up here of 0, and a log2 standard deviation starting value of 0.7. And for those of you familiar enough with Excel, if you highlight those 3 boxes and grab this little box with the crosshairs at the bottom, you can use that to drag the values to the other columns without having to enter them all physically. Right, so we've got all that. Now the magic happens. We press the solve button, and down here we see the solve routine going through and working out the fits for each of those columns, deciding that the second column gives the best fit, and the results for that column are taken back to the sheet, and there are answers available. There's a lot more information than you might normally think about available here, but so I've tried to put in anything or answers to any question that people might ask. And here the really important point is that there isn't one ECOFF. On here there are 5. Because in order to understand what you're doing, you are fitting a log normal distribution. And a normal distribution, a log normal distribution, do not have a beginning or an end. There is no automatic end to a normal distribution. It actually, the upper end is infinity. So instead, we have to choose what proportion of that wild type population we will accept to set our ECOFF. Is it 95% of that population, or 99%, or in the bottom case, even 99.9%? And you do end up with different values. You can see that these are the exact values calculated, but because we are used to the 2-fold dilution series, so these are then rounded up to the next 2-fold dilution. This column tells you what proportion of the population that you have here, the whole population, is actually above that ECOFF at this particular percentage. And also, for the purposes of eyeballing subsequently, what proportion of isolates are sitting at that nominated ECOFF. And you can now see the ECOFF here is either 2 or 4. We can copy those results and find them here. They are in the 3rd row, because I did it once before the first session, and I've done it at the first session, and here's the same results generated on the second session. You have a permanent record there, but we can also go back to the data entry sheet and copy the results, and this can be then pasted into anything, like I usually paste it into a Word document for storage. A small trap, not yet fixed as far as I know, is that this doesn't work on a Mac. For some reason, the graphics don't grab all of the information that you see in this green box, but I don't know, one day that will get fixed. If you're doing this on a Mac, you have to use the Mac's snapshot technique, which is a complex formula of Command 3 or something or other. I keep forgetting because I'm so used to doing the PC. But that means you've got a permanent record not only of the results here, but also of the graphs, and that can be useful for later reference. So that's one choice. I'm now going to get rid of all of that by clearing it all, pressing all those 3 buttons, and then going back and showing you this distribution, which is for Klebsiella aerogenes and ciprofloxacin. So we go through the motions here. And guess there was 30 distributions, and I made, a common Excel problem is that it doesn't store things in the clipboard. So we have to go back and do that again. But here you see a totally bizarre looking distribution because there's obviously a very large number of wild type in this distribution, which is actually an aggregate distribution of numbers of distributions. So with all of those ones, ECOFFinder, the way it's designed, has a problem. And the problem, as far as I can tell, can't be fixed simply. I'll show you how it's fixed, but it can't be fixed simply because it's always looking for the mode. And in this case, the mode is in the non-wild type, it's up here. So what you have to do is take the mode out. And the way to do that is to park the data in this thing called park data area. And, oh, whoops, I'm sorry, I've overcompensated there, but it doesn't matter too much. And then I take that data out so that the wild type mode is the starting mode for this distribution. And that fixes the problem. The technique here is that the data are still available, that they're still visible. When you copy the results image, you still have them. Although I show you can pry them back. So here, having corrected that, we have maybe 250 in the wild type distribution. Start with -5 in that darker blue box and put in the usual 0.7 starting values and fill all the boxes that have orange values above them. And again, click solve. It's got to do 10 columns, so it takes a little while longer. And here you can see the wild type distribution modeled as modeled. But here you also notice when you do the graph this way that this is an example of where the wild type and the non-wild type overlap. So the accuracy of the ECOFF estimation is somewhat compromised. It's impossible to overcome it for all of the reasons we were just discussing about different issues with MICs and MIC variation. But yeah, at least you can see the green line. It tells you where the ECOFF is likely to be, and depending on which percentage value, it's either 0.06 or 0.125 for this particular example. If you really want to, you can bring the data back and put it in the graph. It doesn't change the values, but it just changes the graph. And you can see the wild type following the green curve here is covered. So that's an example. There are other, a third example, but not quite so trustworthy, and I probably wouldn't spend much more time on it. Then I'll just clear the data and put in this data for Neisseria, gonorrhea, and ceftriaxone. This is another common problem that people encounter, that for this particular example, we do not have a complete bell curve. Because the MICs, in fact, go way down to 0.0005, so 3 zeros before a 5, and lower than that, even for ceftriaxone and Neisseria gonorrhoeae. We are trying to model something that doesn't actually have a complete set of values at the left-hand side of this curve, where, if you remember from the graph earlier, we are doing most of the modelling. We're starting with as much data on the left-hand side of the curve as possible. So when we see distributions like this, we always think that the ECOFF is a little less trustworthy than in other cases. So it doesn't matter how good your maths is, it doesn't matter how clever Microsoft is at programming Excel, or how clever algorithms we can come up to analyse it. The output is only as good as the quality of data that you put in it. In this case, we would say we get a value, might be 0.125 or something, but we wouldn't be really trusting that data unless we had many more values sitting below 0.002. So with that, I will stop sharing the screen and be happy to answer questions.
Thank you, John. There's one question in the chat that you could address. Can you get any measure of the goodness of fit of the log normal distribution?
Yes, you can. In fact, if you go to the original paper, there were a number of different statistical techniques and statistical tools used to try and get a goodness of fit. The goodness of fit won't help you decide on the ECOFF, unfortunately. And the other important thing to understand is there is an approximation, because if I had a choice, we'd all have log 2 dilutions and all the half steps in between. The number of data points we got to try and fit a curve is less than optimal for curve fitting. It's usually only 5 values, sometimes 6. And the people who fit curves for a living will tell you, yeah, that's okay, but it's not great. But none of us are going to start doing half-step dilution MICs anytime soon. So we have to kind of learn to live with the quality of data that we have. And one day, a different idea about how to measure MICs will come along, but none of the brains on the planet that I'm aware of have come up with them yet.
Right, there's still time for 1 or 2 more questions. Anyone? If not, thank you so much, John. And thank you everyone else for joining. And again, apologies for leading you astray. It wasn't on purpose. It won't happen again. As I said, you can take in the whole webinar on the EUCAST webpage. in a few hours. I will put it out there. So thank you all, and see you soon again. And have a good day. Bye for now.
Bye.